Welcome to all for this session. Today's class is discussed about female gametophyte or embryo sac. The female gametophyte is commonly called embryo sac. So both the terms are same. So the development of female gametophyte include two processes. Number 1, megasporogenesis. Number 2, development of embryo sac from haploid megaspore. Let us see one by one. The first, megasporogenesis. Megasporogenesis means megaspore formation. Megasporogenesis means formation. See how the megasporogenesis occurs. Where it occurs, we'll discuss. See, in previous video we discussed about the anatrapous ovule and their structure. Now, in this anatrapous ovule, the central region, there is a mass of cells. They are diploid. That region is called as a nucellus tissue. In this nucellus tissue, where it is present near the micropylar end, the special tissue that is called as a microspore mother cell, that is diploid in number. The microspore mother cell is developed into megaspore. Microspore mother cell is developed into megaspore by meiosis cell division. So how will we define microsporogenesis? The formation of megaspore from MMC, megaspore mother cell by meiosis, is called as megasporogenesis. Are you clear the definition? Okay, how the process will occur? First, the mother cell, megaspore mother cell, that is MMC. Megaspore mother cell, it's a diploid in number. It's a megaspore mother cell. See the diploid in number. It has a dense cytoplasm. Dense cytoplasm. Prominent nucleus. And large cell is a large cell, that is MMC. Now the MMC undergo first meiosis cell division, meiosis or reduction division. Then it form diode. Diode means two nucleus of megaspore. So this is called as an megaspore diode. Diode means two nucleus. Then again, this megaspore diode undergoes transverse division. It forms tetrad, megaspore tetrad. Usually, the angiosperm, the megaspore tetrad is a linear tetrad. We call it as linear tetrad. Most of the angiosperm produces linear tetrad megaspore. See, when you are drawing this diagram in megasporogenesis, you should mention chalazal end, and this is called as a micropylar end. This is chalazal micropylar end. See, in the linear tetrad means 4 megaspores are present in it. All the megaspores are haploid in number. Okay. Out of this 4 megaspore, the first 3 megaspore that is present in the chalazal region are degenerated or disintegrate. Disintegrate means disappear. or degenerate, disintegrate. The remaining only one microspore which is present in the micropylar region becomes functional megaspore. Are you clear? Only one megaspore is a functional. From this megaspore only the embryo sac will develop. So, this type of development, embryo sac development, is called as an monosporic development. Monosporic development. Why it is called as a monosporic development? Because in the megaspore tetrad, the 4 megaspore tetrad, among the 4, 3 of them are disintegrated. The remaining 1 megaspore becomes functional megaspore. From this functional megaspore, embryo sac development will be occurs. Okay, that's all about megasporogenesis. Here, what kind of cell division occurs? Meiosis cell division. Which cell is involved here? Megaspore mother cell. Megaspore mother cell. What about this? What about the chromosome number of the MMC? Diploid in number. First, what will form? Megaspore tetrad. Again, it undergoes division. It forms a linear tetrad. Among the linear tetrad, all these megaspores are haploid in nature. The 3 megaspore which is present near the chelacil end is disintegrated. The remaining 1 become a functional megaspore which is present in micropylar region. Okay. Next we will come to this development of embryo sac. That is the second process in, what is that? Female gametophyte. Okay. Here first the functional megaspore. Here it starts with the functional megaspore. What about the chromosome number? Can you say, children? Haploid in number. This is an haploid megaspore. Haploid megaspore. It is the first stage of embryo sac. Okay. Then this haploid megaspore undergoes mitosis cell division. So first mitotic cell division. It produces two-celled embryo sac. Two-celled or two-nucleated embryo sac. Two-nucleated embryo sac. Again, it undergoes division, mitosis. It produces 4 nucleated embryo sac. 4 nucleated. The 2 nucleus is moved away to the move towards the chelacil end, and the 2 nucleus move towards the micropylar end. Okay. Then again it undergoes mitosis. Then it forms 8 nucleated. 4 nucleus in chelacil end, another 4 nucleus in micropylar end. Okay. The 4 nucleated embryo sac undergoes mitosis cell division. Then it becomes 8 nucleated embryo sac. 8 nucleated embryo sac. Okay. Children, when you look at here, this division, 3 mitosis will occurs. This is called as an karyokinesis. What do you mean by karyokinesis? Karyo means nucleus. Nucleus consists division. Okay. Here the nucleus is divided, right? 1 nuclei is divided into 2. 2 nuclei is divided into 4. divided into 8. Okay. So in this step, what will happen? Karyokinesis occurs, that is nuclear cell division. How many nucleus cell division occurs? 3 cell division occurs. All the cell divisions are mitosis in nature. Okay. Then after that, this 8 nucleated embryo sac, the 8 nucleus are arranged in a particular order of this embryo sac. How they arranged? The 3 nuclei, see the 3 nuclei moves towards the chalazolant. The 3 nuclei move towards the micropylar end. And the 2 nuclei move towards the center region or middle region. Are you clear? Up to this step, the karyokinesis get over. Next, this 8-celled nucleoside, 8 nucleated embryo sac undergoes cytokinesis. Cytokinesis means division of cytoplasm or cell wall formation. See, in embryo sac, see in chalazolant, 3 nucleus is there, right ma'am? So around this nucleus, cell wall formation occurs. I said cytokinesis formation occurs. It occurs in chalazolant. In micropylar end, the 3 nuclei also present. Okay. Around this, cell wall formation occurs. Okay. Now cytokinesis occurs. In middle, the 2 polar nuclei arranged in the middle region. So out of 8 nucleus, the 6 nucleus is surrounded by cell wall. That is called as an cytokinesis process. Are you clear? See, in this microchalazolant, this is a chalazolant, the 3 cells are grouped together or 3 nucleus are grouped together is called as an antipodals. Don't confuse it. Anterior region, antipodals, that is present in chalazolant. Then come to this micropylar end, one large cell that is called as an egg cell, that is a female cell. The remaining 2 that is called as an synergids. Okay. See, in this synergids and the egg cell together called as an egg apparatus. Are you clear? What is egg apparatus? Egg cells and synergids, which is located in the micropylar end of the embryo sac, is called as an egg apparatus. In middle, what is that? Polar nuclei is there. Then, in the synergid, there is a filiform apparatus is present. There is a cellular thickness is present, that is called as an filiform apparatus. What is the function of this filiform apparatus? It guides the pollen grain. It guides the pollen tube. Pollen tube into the synergids. See children, near the synergids, there is a apparatus that is called as a filiform apparatus. There is a cytoplasmic thickness present in the synergids that is called as a filiform apparatus. This apparatus guides the pollen tube into synergids during fertilization. Okay. The next 3 antipodal cells. So, here the embryo sac development. See, the embryo sac is 8 nucleated embryo sac. 8 nucleated and 7 celled. 7 celled. Can you count it? 8 nucleated. See, 3 antipodals, 3 nuclei, 2 polar nuclei, 5, 3 plus 2, 5, 1 egg cells, 6, 2 antipodals 8. So 8 nucleated. Are you clear? The next one is called as 7-celled. 7-celled 3 antipodals, 2 synergids 5, 1 excel 6 and the whole cell, that is 7. So that is called as 7-celled. Example, polygonum. See, in polygonum, the embryonic development or development of embryo sac is 8 nucleated and 7 cell. 8 nucleated and 7 cell. And one more thing, this embryonic development is called as an monosporic development. Why the embryonic development is called as a monosporic development? Because during this megasporogenesis, the linear tetrad, among this tetrad, three of the megaspore is disintegrated, one becomes functional. One means mono. Only one megaspore becomes functional megaspore. From this functional megaspore is developed into embryo sac. So, it is called as an monosporic development. Now, what and all develop in megasporogenesis? Megaspore formation, then embryo sac. That is a female gametes get ready for pollination. And in microsporogenesis, microspore formation, that is pollen grains are get ready, right? So now the both of them are get ready for pollination. What do you mean by pollination? Transferring a pollen grain from anther to stigma. That is called as an pollination. Okay. Then the next video we will learn about pollination. Thank you.